Hi, how are you guys? This is Ömer Gökçümen, and I am an associate professor in the Department of Biological Sciences at University at Buffalo. And I'm going to talk about some of my research today, and hopefully I will also give you some idea about evolutionary genomics and how it relates to biomedicine. So to start with, I just wanted to tell you that I'm an anthropologist by training. My PhD is in anthropology, and my undergrad, however, is in molecular biology. And my postdoctoral work was actually in a medical school in a pathology department. Even though these different departments, different interests seems very disparate, actually they have a lot of commonalities when you think about this one question, which is what makes us human? Because, being a human is a very general, very complicated thing. And it's a subjective question, if you think about it, because we are not that interesting when you think about certain things. For example, we are not that interesting when you think about genome size, because our genome is kind of medium size. We are not that interesting if you're thinking about who runs the fastest. We are not that interesting when it comes to the densest bones. We are only interesting when it comes to complex culture and maybe thinking about self-domestication, where we actually change the environment so much that our ecology is changing. And we're also interesting because when you think about it, because it's us, that we are actually asking questions about us. So generally, anthropology and the question of what makes us human is a subjective one. So one of the difficult things in studying humans, as I do, is to take all these different aspects, because I'm already familiar with the molecular biology of what makes us human, the cells and the molecules and the proteins. I'm slightly, I'm familiar with the anthropology and anthropological research. I'm also know pathologies and diseases and how they work. And this is, even those three major components are only a small part of what makes us human. What about politics? What about culture? What about language? And so it is very difficult to put everything together, so maybe impossible. And like I said, it's a subjective issue. So it's another issue, is that to make sure that you find some leverage, some methodological leverage, to make sure that you're looking at the question from an objective lens. That's how I think about that problem. And one of the great things that we have, great tools we have, is genomics, the genes. And the reason why this is interesting, I will tell you, there are a couple of reasons why they are very good tools. The genomes are very good tools to understand humans. One is that it will give you kind of an objective look, a relatively objective look at human variation, disease, and humanness. For example, here I'm showing you two chromosomes. Some of you may already recognize them. This is an X and Y chromosome. Probably you already know that the smaller one is the Y chromosome and the bigger one is the X chromosome. So if you think about it, and if you think about aliens coming to the world and have no knowledge about history, etc., they come in and they just want to understand humans, and they look at our chromosomes and realize that, like there's two sexes, and which one is the more important one? And they realize that, like X chromosome is the female and male chromosome the male. Obviously, they will immediately realize that the females are more important of the sexes. And that is clear because they have two X chromosomes. X chromosome is much bigger. Y chromosome actually comes later and is only a tool to manipulate the other parts of the genome so that a male can be a male. So what I'm trying to say is that you can use genomes to cut through a lot of historical cultural narratives to find new, innovative, and slightly more objective ways in which you understand humans. So the other thing that is really great about genomes is that it will give you an idea about the inheritance of different traits. Because as we will see in this lecture, and I'm sure that Josh and Sam talked about this a lot in this class, that almost everything, all the traits that we care about in humans are controlled partly by the environment and partly by some biological inheritance molecule, and that inheritance molecule is the genome. So it's passed down from generation to generation, and the variation in it determines genetic basis of all the traits that are heritable from generation to generation. So the good news is that sequencing genome and understanding genomes becomes really, really cheap. So when I finished my PhD and published my first paper, I actually used about 300, I collected 300 plus Altai and Kazakh samples. And that was very, a lot of samples at the time. And I looked at the mitochondrial DNA variation. It took me literally years to get all the data, analyze the data. And I created this tiny little dot amount of data, that red dot. And literally 1 year after that publication, I started my postdoc. And for a single Korean genome, we generated thousands of folds more data for just one individual. And the reason why that jump happened is there was a revolutionary jump in the technology, where maybe you already heard about this next generation sequencing technology becomes available. So it was much cheaper and easier to conduct such bigger data production. And now, we just published this paper 2 years ago, I think, and we easily generated, this much data from my lab. My lab is not that big, and that was an easy project. And now we can use the similar technologies and even better technologies to generate even more data. Go into ancient genomes, go into sequencing of the RNA, sequencing of the parts of the genome that binds to specific types of proteins. So this omics technologies become so powerful that we meaning that all anthropology labs, biomedical labs, biology labs can use those kind of data sets to come up with integrative analysis of genomes, transcriptomes, even proteomes to compare and understand humanness, human uniqueness. The problem is that when you have that much data, guys, it becomes what I call is Library of Babel problem. This is a reference to, it's kind of a dorky reference to Borges's short story, where he describes a library where all the information of the universe is available, but there are no index cards. So even though in this library, you literally can have answers to any questions that you have, you actually have zero answers because you do not know where to look at. So genomes, transcriptomes, all these omics technologies that generate mountains of data is similar. Yes, it generates all this data, but you do not know where to look at for understanding a disease, for example, or understanding why, I have no hair on my head, which bothers me a lot nowadays. So then what the problem actually is answerable in the sense that we do have evolutionary theory. So that is a very powerful framework to get the data, ask specific questions about human variation and human uniqueness, and guide through the data sets, analyze the data sets in such a way that we can actually have specific answers to specific questions. Without that, at least my laboratory would be lost. And I will give you some examples that how it will help in understanding human disease and variation. So one of the ways, for example, that we can easily think about is, comparing our genomes. This is my son Evrim, and comparing to other monkeys, for example, other primates, and in this case, monkeys, we can actually ask the question, well, like, what are the differences in Evrim's genome and these monkeys' genomes? And you would be surprised that how similar our genomes are with these monkeys, like, more than 90% in most cases. And the differences has to harbor all the differences, inherited differences that makes Evrim a human being and these different types of monkeys. So this is like one type of work. Another thing that is really obviously important, but that is what makes us human, is not just the differences between us and the monkeys that we can actually look at, but also the variation among us. That is part of who we are. There was never a time in our past that we were not diverse. There was always diversity in our species or in all species. And that variation is carried with us, changes over time, but is with us. So there was no an original human. There was no specific homogeneous population that makes us that are our ancestors. There was always that diversity, and that is a fascinating aspect, an important aspect of humanness. So with that, I think I want to wrap up the first part of this lecture, where what I'm trying to say is that an evolutionary genomics laboratory like mine ask questions about human nature and variation using omics tools. I think I was using genomics tools, but at this stage, we are using we don't actually look at only genomes. The tools are available and cheap that we can actually look at lipidome, where we look at all the lipids in the cells and tissues. We look all the transcriptome, meaning that we look at all the RNA molecule and their sequences in a given tissue or an organism. We look at proteomes, etc. So we are actually in the microbiome, which we actually try to link to genomic variation, for example. So we have all these omics tools, and we actually generate data and then ask questions about humanness within an evolutionary framework. And that provides us really interesting angles. So within that context, my lab, because one of the things that Josh asked me to do is to tell you how I think about the problems and how I actually construct my projects. That's why I wanted to share this slide with you, where we are looking at different aspects of, like I said, human variation. And specifically, we are trying to combine genomics, anthropology, functional genetics, and health all together within an evolutionary theory framework. And we basically publish many papers in different combinations, but I will give you one example on growth hormone receptor in this particular lecture. But before moving into that, I want to basically ask a more specific question that we can answer with the tools that I described. Why do we have diversity in the first place? Why do I have no hair in my head? Why some of us are darker and some of us are lighter? Why do we actually, some of us are taller and some of us are shorter? And in that regard, why do we get sick? Why do we have genetic variants that lead us to susceptibility to cancers, autoimmune disorders, allergies? Why do we have them? And that is actually a question that even Darwin asked hundreds of years ago. It's a fundamental question in evolutionary biology, because if there's one optimum, meaning that if something is so adaptable in one place, then every organism from the species should be reach that optimum and stay there, right? That, for example, if Idris Elba is the epicenter of the optimum of the human males, that's the best, most fit, most evolutionarily well adapted individual, then why are we not all looking like Idris Elba and there's all this variation? So the answer to that question is actually not an easy one. Let's actually then, before going into more into that, let's actually think about how traits, different phenotypes, different biological aspects of humanness or any organism is determined. And very simply, it's determined by genetics and environment. So you can take anything, from eye color to skin color to hair distribution to like the size of your liver to the molecular markers like diabetes markers or blood markers for cancer. All of this is determined by genetic factors and environmental factors. In this chart, you're seeing that on the x-axis, you are seeing it's a schematic presentation of that idea. On the x-axis, you have the contribution from environmental factor. Like if you go from left to right, you have increased effect of environmental factors, and from top to bottom, you have increased contribution of genetic factors. And if at the environmental factors, it's just like, accidents, right? You basically do something silly. I basically went to the roof, by the way, last week to clean the gutters. And it was a very bad idea because I got trapped there. It was I could not get down for a while. So that was kind of dangerous. And if I fell and, die from it, that may be an accidental injury, and it's nothing to do with my genetics, to most extent. Like it's very little to do with my genetics and a lot of things to do with, that I have a roof, cultural reasons, like, maybe I was actually not thinking straight that day, whatever. And on the other side of the spectrum, that you have monogenic diseases. These are mostly Mendelian disorders that are often, unfortunately, very bad. And they actually lead to very short lifespans, often infertility, either because, you don't have the proper sperm or the egg production, or that you don't actually live enough to be sexually active. But either way, that these are pretty serious stuff that actually is completely, it doesn't matter if you eat the best food, if your mother has like the most healthy pregnancy, if you have the genetic factors, often comes as recessive. These are recessive alleles. And if you actually somehow you're unlucky enough to have one of the faulty allele from the mother and one from the father, then you have the disease no matter what. And in the middle, and that is the majority, is complex diseases and traits, where genetics and environments work together to create the end results. These actually include, like I said, from your height to how chubby you are, to cardiovascular disease, you're susceptible to the cancer, type 2 diabetes, pretty much everything that is variable among us in this class is determined by a combination of genetic factors and environmental factor. And it is not easy, as you will see, to disentangle them. But let's actually think about how evolution is shaping the alleles, the genetic factors that affect those traits. And if you look at it, one of the way in which you look at genetic variations among humans, there are millions of them, tens of millions genetic variants among humans. And when you look at it, one way to do it is that on the x-axis, I'm showing how rare or common these genetic factors are. And on the y-axis, I'm showing how much they affect a trait. And after years, we did not know anything about this, by the way. It's just a matter of the last 10 to 20 years that the empirical data show that, that we actually have some understanding, thanks to the empirical data that we generated, we have some idea how they're distributed. We now know that the easiest to find is common variants that are like the genetic variations that we share, among us, maybe 10%, 20%, 30% of us have those variations. And they also have a high penetrance, meaning that high impact, high effect on a trait. If you have that, for example, height is a good example. Imagine a situation that if you have an allele, a change from A to T, for example, in one part of your genome, and boom, you're 5 inches taller. And if you don't have that allele, you have 5 inches shorter. That is not the case, right? Like, almost there's very, very few alleles that are like that. Some of them that comes close to it is BRCA1 mutation, for example, which, as you may know, Angelina Jolie actually removed her breast and her ovaries because there's like, one allele that is pretty common in European populations, and that allele leads to a substantial increase of breast cancer and ovarian cancer risk. And it is pretty common in the population. So, but generally they're very rare. They're generally those common and high penetrance guys are not found that often. And you have on the other side of the spectrum, you have very rare guys, but they lead to a very high penetrance, and they are generally Mendelian disease guys, where, like I said, with the very bad muscular dystrophy kind of bad diseases, they are actually on that group. And most of the variants nowadays that we are identifying are common variations that are have modest to low impact on your traits. For example, height is a good example again. Like I said, there are no single variant that makes you like 5 inches taller, 5 inches shorter, etc. But there are literally hundreds, more than 100 variants across the genome. And the combination of those will actually lead to much taller or much shorter heights. For example, if I have all the quote unquote right alleles for height, then I will be predetermined genetically to be much taller. And so my range would be depending on what I eat and how, like, what kind of environment that I grow up in. Let's say that I will be 6 feet to 6.5. And if I have like the other end of the spectrum, I have variants that makes me much shorter. It's basically a combination of 100 different variants to work perfectly in concert that will actually lead me to be much shorter. Maybe I will be 5 through to 5'6", depending on the environment. So it's not just genetics, but it will give you the baseline for how tall you will be. And if you change one of these variants, it will affect your height significantly, but only a very small amount, like just maybe 1/4 of an inch, 1/5 of an inch, etc. So this is an important part because it really, this chart, even though it's a simple chart, it really determines how we envision genetic studies. For example, a lot of effort and money and resources goes into low frequency variants with intermediate penetrance right now. So in that middle part, that's why we have now gigantic cohorts reaching to 1 million people, where we actually try to identify, say, the diabetes susceptibility, so that we can identify low frequency variants with intermediate penetrance. So thinking about evolutionary framework, by the way, we can actually start thinking about really rare variants, for example, rare variants with high penetrance, high effect. And this is actually an example, which is a Carter-Genner syndrome. It can be a pretty dangerous syndrome that actually leads to, that affects the lungs and breathing and mucus formation, etc. what is interesting is that there are actually a particular genes where the amino acid sequence is really, really conserved. So you're actually looking, let's actually first look at the right side. So we are looking at Homo sapiens, which is humans, mus musculus, which is the mouse. Then you have the rario, which is the zebrafish, and you have the worms and the bacteria, etc. And you have the same amino acid sequence or very similar amino acid sequence, definitely in all animals, but even with other organisms. So it has been conserved for a long, long time. That means that, don't touch something if it is working. Like, that's the idea, right? Like, if a motor is working in a car, you don't actually poke and, get a nut out, or change, or put something in the oil or something like that. You basically leave it if it is working properly. So it's the same idea that if it is not, because if you change it, then probably the organism either dies or have difficulty in reproducing. In cases where there's an issue, any change, where people actually show in these locations that in the patients, they actually see that there's a G to A change at the nucleotide level, which leads to this E to become a K. Same here. Sometimes it's not this amino acid change, but you have another mutation that is already also recorded, where this A becomes a V amino acid. And what actually ends up happening is that you actually have a pretty serious syndrome, pretty serious sickness. And people actually use this conservation, which is an evolutionary framework, to figure out of hundreds of thousands or millions of different variant that individual has, which one is the one that is actually leading to this particular syndrome. Because they look at all the variations that changes otherwise very conserved amino acids. So going back to this chart, if you look at this part of this chart, then what you need to do is GWAS studies, because it's not as simple as finding a single variant. It is about finding multiple variants affecting the trait, and the effect is not perfect, right? Like, because with the Carter-Genner syndrome, you actually have 100% penetrance, meaning that if you actually have that change from E to K, you do have the syndrome. In complex diseases, it's not just one variant, one allele, it's like multiple alleles. So it's not like, when you actually look at all the patients with diabetes or heart disease, you actually see that every single one of them actually have a particular variant, and everybody without the heart disease have another variant. That's not how it works. Instead, the proportion of alleles will be different from people with heart disease versus people without heart disease. And that is it's exactly what an association study is. You basically take, say, 1,000 cases with heart disease and 1,000 people, 1,000 controls without heart disease, and you look at if an allele, the frequency of an allele is different between cases and controls. And if it is significantly different, then you actually say that the C, which you see much more in the heart disease cohort, may be actually associated with heart disease. So what you can do is that you can actually do this nowadays, not just by one allele like this, but rather for millions of alleles at the same time. And you can actually look at the genome-wide association between heart disease and, let's say, heart disease and genetic variation. So you will find multiple hits that are significantly associated with heart disease. Here, I know that it's a little bit early for you to learn that level of statistics, but one of the big issues here is to control for multiple hypotheses, which means that because you're looking at so many different alleles, just by chance, some of the associations look real, some of them look different between controls and cases. However, they are not. It's just because you're looking at so many different alleles, it's just noise. So to cut through it, you require a lot of samples and very low p-values to be sure that what your hit is, the allele that you're looking at is really associated with whatever trait you're looking at. Okay, so far so good. I hope this was clear. And now just to reiterate, iterate what I have been saying is that with complex diseases, there's a weird situation that let's say that you're associated, you actually find, you made a nice well-powered association study and you find a genetic variant that is associated with, say, diabetes. So, and you basically look at your own genome and find that you do actually have that particular variant that is associated with heart disease. What are you going to do? Is it means that you will have heart disease and you're going to die early? Or does it mean nothing, that you shouldn't worry about it? So we still do not know much about it because there's a big black box, big question mark, because we know that there's a genetic variant is associated with disease, but sometimes magic happens and there's no disease. Sometimes magic happens and there is disease. It's just that we know that if you have a genetic variant, the occurrence of disease is elevated. So that in between, how come a genetic variant leads to the RNA, and that RNA leads to a change in protein, or maybe it's a regulatory thing, and then there's a protein dosage change, and that leads to some changes in a cascade in a signaling pathway that leads to changes in your kidney function, which leads to different uric acid in your bloodstream, and that actually leads to some sort of disorder or not, depending on what you eat. Like that kind of mechanism is very difficult to reconstruct, and it is the golden egg, if you will. It's like the gold that we want to find in these kind of evolutionary genetics studies. And if you basically actually look at an autoimmune disease, which is a complex disease, and this, I like this because this is a nice chart. There are multiple genotypes, gene 1, gene 2, gene 3, gene 4, that is affecting the abnormal innate response, decrease in thymic expression of antigens, reduce activation threshold of cell, reactive lymphocytes, hormone levels, all sorts of things. Then you have the lots of environmental factors. For example, what are you exposed to? Are you exposed to COVID-19? Are you exposed to flu? Are you exposed to, I apparently had lots of parasites when I was growing up, and which was actually pretty horrible when I was growing up. But now it is possible. Some people claim that it is because I have the parasites. I actually don't have allergies. I don't know. And again, environmental factors like, what you're eating, pregnancy, how hot it is, may all leads to the presentation of the disease and how severe it is, etc. And of course, once you have that complex autoimmune disease state, like if you have psoriasis, for example, or if you're asthma, then you can still, even then, how that is affected, how it's actually present itself is different from individual to individual. Some people can, have normal lives. Some people actually have to be intensive cares. Okay, so that complexity, unfortunately, is where the bottleneck is. That, right, there's a lot of effort and resources and thinking goes into understanding how the known genetic variants associated with diseases are interacting with the environment to lead to complex traits. It doesn't have to be a disease, but complex traits. Okay, so now let's talk about my research, which is all about this whole idea understanding these kind of complex traits, but with an evolutionary framework, with an evolutionary look. And the way in which we imagine this, guys, is that we look at all the variations in the genome. We then think about, okay, what makes us human? Like, what contributes to the diversity? Then we actually come up with evolutionary scenarios that we think about that is known from archaeology, from other anthropological research. For example, we think that balancing selection, which maintains variation a long time because, because you need to fight so many different pathogens. So like, those genetic variants should be good for one pathogen, but bad for the other. And so all of these different types remain in the population, for long times. That is a scenario for balancing selection. If that is your question, then we come up with an algorithm, a mathematical way, a pipeline to reduce all the variations and get rid of everything that doesn't fit to balancing selection scenario. Then we basically, for the candidates, we come up with an omics search for function. We look at lipids, we look at RNA, we look at proteomes, we look at all the genome-wide association studies and see if there's an enrichment, it's a functional relevance of the subset of variants that we come up with. Then based on this, combined, we come up with the biological hypotheses, and then we come up with local specific analysis to determine exact mechanism and function and the evolutionary history of these variations. So we do exactly that for this project. And one of the things that we have been looking at among all the variations is that instead of just single nucleotide changes from A to T, 2 to C, what have you, we figured out, a long time ago, not just us, but like, other people as well, that there's all these structural variants, meaning that entire sequences are deleted in some individuals and present in others. Sometimes you actually have mobile element insertions that are present in some people and some it doesn't in others. There are gene duplications that actually have, like, some people have 5 copies of a gene and some other people have 4 copies of a gene, etc., etc. So it's not that important what are the exact types, but we were interested in these type of variations. And one of the things that we look at is these structural variants, particularly because structural variants make a good portion of the variation between 2 different humans. There's actually only 1% of the base pairs differ between 2 human genomes, and more than half of them are actually structural variants. And we argue in my laboratory and in other laboratories now that genomic structural variation may be underlying a lot of the differences between you and me, as well as a lot of the differences between me and a Neanderthal or a me and a chimpanzee, for example. And within that context, we already narrow down our search space from like all the variations in humans, which is like, millions of SNPs and structural variants and what have you, to tens of thousands, where like, we are only looking at deletions, duplications, etc. And still it's a, kind of a needle in the haystack. But now the next step is to look at balancing selection stuff that, like I mentioned before, that one of the projects that we had in the lab is to figure out if there are structural variants, these deletions, duplications, that behave in such a way that fit balancing selection. And to give you an idea about what balancing selection is I think the best case is sickle cell mutation in humans. If you have the sickle cell allele, in fact, there are multiple sickle cell alleles, but if you have the S allele, normally, if you have it homozygously, it actually leads to a pretty painful, pretty detrimental disease called sickle cell anemia. In fact, in US, for example, it's not that uncommon to have sickle cell anemia, especially common in people of African descent and people of Turkish descent, for example, so it's not that uncommon. However, what the community realized since 1950s, 60s, is that sickle cell anemia is actually much more common than it should be. It should be eliminated from the population because it's bad for you. How is it maintained? It comes to our first question that we have been asking in the beginning of the presentation. And the answer to that is that if you actually have that allele, even though it gives you a risk of sickle cell anemia, obviously, it protects you against malaria parasites. It actually gives you a very high immunity. So, and if you actually have this allele heterozygously, meaning that one of your chromosomes, say from your mother, has the S allele and the other chromosome have the normal allele, then you have the best of both worlds, because your anemia is very, very mild. You probably won't be going up to Mount Everest, but you wouldn't even notice if you're living a normal life. Plus, you are protected against malaria to a large extent. And so the mortality for if you're living in a place where malaria is common, if you have the heterozygous situation, that you have the mortality very low. And the result of this, guys, is that this allele is lingering in the population for a long, long time. And that is called balancing selection. So then we basically want to go back to our funnel here and say, okay, we already reduce this all variations to just structural variations. And then we want to further reduce that. I want to know if there's any stuff that, any structural variants that fits balancing selection. And what we actually end up doing is to look at Neanderthals and Denisovans, their genomes, which are now available, and see if there are actually any deletions that is polymorphic among us, but it is also shared with Neanderthals and Denisovans. So this is one example where this particular gene, this region is deleted in, I think, 40% of the humans, but is also deleted in Denisovans and Neanderthals. So you can actually see that these are sequences coming from this individual mapping to the reference genome, and you can see that nothing is mapping to here. That means that there's a deletion in this region, same here. So we are actually sharing this deletion with the Neanderthals and Denisovans. So we actually come up with, using this, we come up with hundreds of deletions. And what it does, guys, that when you actually look at this sharing, we can actually then say, okay, these are the deletions that are human specific, and they are not good candidates for, they're only seen in humans and not shared by archaic humans, Neanderthals, because they are new and they not necessarily be that old. They're not been maintained for a long time, which is a hallmark of balancing selection. But if you actually look at these purple guys, which are the guys that are shared with Neanderthals or Denisovans, and as a consequence, what it means that these purple guys have evolved before humans and Neanderthals are diverged from each other. So essentially, some sort of a Homo erectus-like group, our ancestors, our ancestral population already carry that variations, and those variations never went away. So we wanted to know if these are actually potentially balancing selection. And one of them, we actually did these omics categorization and look at all the functional relevance of these different variants. And we find one particular deletion that is really interesting, and that is growth hormone receptor gene. And the reason why this is interesting, guys, is growth hormone receptor looks as if it looks very similar to the gene that we looked at for the this gene, where the Carter-Genner syndrome, where the protein sequence is extremely well conserved. And if you think about it, that makes sense because it is the growth hormone receptor, right? Like growth hormone is obviously important. It makes us grow, it makes the cells divide, and its receptor is also important. Thus, it's conserved pretty much in all mammals. It has very similar amino acid sequences. And somehow, this part is missing in half of human genomes, and it is been missing for at least 1 million years. So what gives? How is it possible that such an important conserved genome where, like, most humans and chimpanzees and even the anteaters actually have the same sequence, and this part is missing in half of human genomes? So we were actually very curious, and at the molecular level, I don't know if you are taking cell biology, but the growth hormone receptor, which is this this transmembrane protein, is sitting in the middle of a very important cascade, which leads to growth and metabolism. This is by the way, this is the dimer, receptor dimer, and the blue guy here is the growth hormone binding to it. And when it binds, it's almost like a switch. It basically activates these downstream cascades. So the first question we asked then, okay, is it really balancing selection? Is it really evolving neutrally? And one of the things that is good about being a PI is that you actually don't do any work. You play video games and you let other people do your work. That's not true. But I'm actually emphasizing all the people who contributed the work, because as you will see, and as you can guess, that these projects are big enough that it will require multiple people working at the same time on different aspects of the project. So my my ex-postdoc now, she's a faculty in Norway. She actually looked into this. And one of the things she did is first look at how common the deletion. And you can actually immediately see that the deletion is very common pretty much in everybody, but it is actually even more common in African populations. It's the major allele. The blue is the deletion. And we looked at multiple Neanderthals and some humans, etc. And we can see that all Neanderthals we looked at and the Denisovan individual is deleted, and some people actually have the deletion and some people don't have the deletion. And the first thing we did, guys, is we look at if there's any selection acting on the haplotypes. And we basically found, to our surprise, that there's selection against the deletion in the Asian population. So this is actually a tough graph to tell you about, but I think I will tell it anyways, because it actually gives you a kind of a flavor of the math and the way in which we approach these ideas. So the idea here is that if you can look at the allele frequency change of the deletion over time, and you can do that by looking at the overall genetic variation around that deletion, then you can guess what is the change from one time point to another. And if that change is so unusual that you cannot really explain it by just random chance. And that is what we did. And what we actually found is that in Asians, in East Asians, it seems that this particular haplotype, this particular deletion was maybe more than 80%. And it decreased very rapidly to 15% in less than 30,000 years. And that is pretty remarkable, and we cannot explain it by chance events. And what we actually end up having then is a plausible model where we know that the deletion actually increased in our ancestors, in a Homo erectus ancestors, I guess, to almost to fixation, meaning that pretty much everybody among our ancestors have the deletion. And Neanderthals and Denisovans, actually, I think all of the individuals from those populations have the deletion. And until very recently, that deletion was very, very common in the human populations. And only recently, in some populations, it decreased, and it decreased very rapidly. And if you look at it now, is that in archaics, the yellow is the deletion, red is non-deletion. And you can see that pretty much all Neanderthals, everybody has the deletion as far as we can tell. In Africa, it is very common, and it is less common in Europe and Asia. So then, okay, that's fine. This does not fit our sickle cell model, by the way, right? It is actually different because in sickle cell, it seems that as long as you have malaria, you have both of the alleles to remain at the same frequency for a long time. It seems that it's a little bit different where the reason why we are seeing this very, very old variation is because it was beneficial early in our evolution. And then it is not that, it is actually out of favor, if you will, in the last 50,000 years or so. So it's basically a change in the adaptive pressures over time that leads to what we are observing today. Because if the adaptive pressure has been the same, and it's better to have the deletion in all conditions, then we should all have the deletion now. Like it should be a human specific thing, like, bipedalism. However, that's not the case. Some of us have the deletion, some of us don't. So one explanation based on our data is that it was beneficial at some point, but then it fell out of favor, evolutionary favor, and now we are back to going back to our ancestral state in a way for that deletion polymorphism. So that is great and all, but what is the fitness advantage conferred by GHRD3? And that is, done by my ex-PhD student, now a postdoc at the medical school at UB. And what we actually first do is look at the associations. Do you remember our associations, like, where we actually look at the controls and cases and see how that affects these different traits? And this is exactly what we looked at. Both we reanalyze some of the data, but also look at the existing literature. So we basically find small birth size, small placenta size, time to sexual maturation, which is faster if you have the deletion, the changes in bone density, changes in metabolic markers, especially among certain type of obesity cohorts, etc. So you actually find all these metabolic and developmental impact of the deletion, but these effects are generally pretty minimal. It's not like a huge change where instead of I think it's affect the birth size, for example, 5%, which is not that small, but it's not that great either. So generally, it's healthy birth. You're just born a little bit smaller if you carry the deletion. So and by just looking at it, it is very hard to make sense of it. Like, why would that be selected for or against? But if you look into the ecological literature, there's actually a very good amount of literature on a particular phenomenon called compensation growth. So the idea is that under bad conditions where like resources are very limited, one strategy that animals basically employ is to born small and then catch up later. And that kind of makes sense. And because I am a little bit goofy, I am thinking about myself, and I'm like, what, 5'7", I'm 160 pounds or so, as opposed to Arnold Schwarzenegger, who is like this huge, or like The Rock, right? So obviously in normal conditions, the rug is better at everything than I am, pretty much. However, if we're in an island, right, and if there's like zero food or like very little food, and we have to like, we have to survive with 1,000 calories each, I'm pretty sure that, Dwayne Johnson like will need much, much more than 1,000 calories. I probably can survive over on 1,000 calories. So this is kind of the idea, and this is kind of the balance that we are talking about. And for the compensation growth, people actually look at all the different aspects, like what do you need and what are the consequences of that compensation, compensatory growth. And when you look at it, it's all about stuff that we already observed associated with the growth hormone receptor deletion polymorphism, bone ossification, growth rate, age at sexual maturation, metabolic markers, lifespan. And the big thing here is the reason why this is important for evolution is that it provides starvation resistance. So then we hypothesize maybe we can actually seeing this situation because starvation was a big deal earlier in our human evolution, and it is not anymore. And if that is the case, it will explain why we actually see it, the increase and later decrease of the deletion. And to actually look into that, guys, we work with our collaborator Neil, and it turns out, and I did not know that, especially protein malnutrition leads to a particular type of metabolic syndrome that after severe malnutrition, some kids actually develop this particular metabolic syndrome, which actually presents itself with this fluid intake and fluid accumulation in the belly area. And apparently it's a major metabolic syndrome that actually requires further treatment even after starvation ends. And it does have a genetic basis. And it turns out that when we look at Malawi children that survived severe malnutrition, the ones that actually the cohorts, the kids that actually survive and develop that particular metabolic syndrome actually show smaller number of the deleted haplotypes, meaning that it turns out that the deletion seems to be protecting. If you have the deletion, it protects you against this particular metabolic syndrome. So in other words, you survive starvation better if you have the deletion. And that is kind of a big finding. It is pretty remarkable, and that we actually find significant results, even if you're looking at relatively small sample sizes. And after that, we want to understand, fine, like, we are getting somewhere. What is the mechanism for this? Like, what is going on? And one of the great things of our times is that CRISPR, which is, as you know, the Nobel Prize last year, is a very powerful tool because it's cheap and it is relatively easy to implement. So we managed to cut the exon 3 in the mouse with this, and we have a mouse. We have a mouse that actually has the deletion, and a mouse which is exactly identical to our deleted mouse, except it doesn't have the deletion. So it's a perfect model to understand what's going on, because you can compare, and these are, you can breed them in identical conditions. They're genetically identical except for this one particular deletion. So you can actually compare the lipids, the growth, the liver transcriptome, etc. And that's exactly what we did. And we, because we are interested in starvation, we basically created male and female cohorts that are wild type or deleted, and some of them eat normal diets, some of them eat calorie restriction. And first of all, we actually look at the lipids. And one of the most amazing things, guys, is that looking at the lipids under both diet, normal diets and color restriction, not only that having the deletion affects the lipid composition in your serum, all the cholesterols and fatty acids and triglycerides, etc., but it also affects in a sex-specific manner. That if you're female, having the deletion, it seems increase the blood lipid content and decreases it if you're male. That is kind of an amazing first look into the metabolic markers. But then when we look at individual genes in the liver, one thing that was really weird, guys, is we found that we found two things. One, under normal conditions, under normal diet, if you look at female, I'm comparing females, which are orange, males that are blue, and we have the wild types and deleted, wild types and deleted, and the left ones are like normal diet, the right ones are color restricted. So if you look at this first gene, you actually see that color restriction really increases the expression of the genes independent of the sex. But in normal diets, it is as if the male mice, in male mice, this gene acts as if there's color restriction. And we find many genes, like I think dozens of genes that are similar to this. And almost all of them are related to circadian clocks. It turns out that circadian pathway is important. I'm not going to go into this rather complicated complicated graph, but I'm going to tell you that based on that, we actually reconstruct a mechanism where we see that the growth hormone pathway in males are affected by both color restriction. So the effect of this deletion in males is similar to color restriction in the sense that there's an already established feedback loop. When you actually starve yourself, essentially what happens is that there's all these circadian rhythm genes are overexpressed, and then it gives a feedback saying that to adjust your growth hormone levels and growth hormone metabolism. So the effect we are seeing is very similar in males if you have that particular deletion. So further, guys, what we actually found is another set of genes that are only differentiated. As you can see, there are sex-specific genes. You can see that under normal conditions in wild types, it's really different from males and females. The females have lower and males are high for this gene, and it's the other way around for this gene. And having the deletion under normal conditions have no effect whatsoever. But under color restriction, guys, that under color restriction, if you have the deletion, the sexual dimorphism in the expression of these genes actually is lost. And again, these are two examples. There are dozens of genes like that. And it turns out that the reason why we are seeing this is that under color restriction, our famous circadian rhythm feedback loop that actually adjusts the growth hormone is not enough, because you have color restriction, which already makes it makes an impact, and on top of it, you have a very similar impact, which is stacked upon the color restriction effect. So you have the deletion plus the color restriction. It overwhelms the compensatory mechanisms, and the result is that in males, it basically makes the expression female-like that's in the liver. And this is a really, really fascinating thing, because this growth hormone pathway is one of the reasons why sexes are different from each other. That you actually, we have the males have certain type of muscle systems, growth, etc. And females have their own way of own metabolism, and growth hormone is at the center of sexual dimorphism, the growth hormone pathway. And what I'm saying now is that under color restriction, if you have the deletion, it seems that males become female-like. It reverts back to its original, because females are probably like the have the more ancestral state, and males evolved to be different from females. And now with the deletion plus the color restriction, you're reverting back to more ancestral female-like state. And the macro consequence of this is that if you're looking at females and males, you see that there's like 30-40% difference in weights normally. And this difference is similar under normal conditions if you're wild type or if you carry the deletion. But if you actually are color restricted, under color restriction, you actually have reduced sexual dimorphism, which is already known, but still this is like more than 10%. So both of them are smaller, and like the whole difference is reduced a little bit. But if you're actually in the carry the deletion, there's no more sexual dimorphism. They're essentially identical in size when you actually under color restriction and carrying the deletion. So this is really fascinating stuff, because this is the first time the clearest example of sex and environment dependent effects of a very common polymorphism in humans. And we think that we have this very new model of situations where it is possible that certain alleles like this deletion actually was very beneficial early on human evolution, but is not that much of a big deal anymore, and thus is going back in allele frequency. And this may be due to modern human behavior. Maybe we have technological buffers, for example. It may be the climate change. It may be the changes in the food availability, but there may be other variants that fits to that. And another thing that is interesting is that this is a fantastic example model for understanding receptor binding. If you're interested in molecular biology of human and mammalian systems, we want to know how does it affect growth hormone binding? How does it affect downstream effects of growth hormone pathway? What happens if we have heterozygous versus homozygous deletions? So but I think more important to this particular class, that you immediately start thinking about, okay, fine, like we basically find this evolutionarily interesting deletion, and it may explain, for example, why intermittent fasting works for some people and it doesn't work some others. It may be that if you have the deletion and if you're overeating, maybe the effects of it is that you're more susceptible to become obese, even if you're actually taking the same level of calories. It is possible that it is affecting the basal metabolic rate, for example. And it's definitely important for sexual development that some of us, I was actually one of the very early boomers, if you will, for puberty. And some people actually have much later in life. And we know that there's a lot of environmental impact on because of the foods and other things that is in our diet. It seems that the age to puberty is moving earlier and earlier due to environmental effects. How that plays out for the within the context of the deletion is an open question. So there's all these ideas that comes when you think about it, because it's a very nice example where the effect of a genetic variant is dependent on the environment. Okay, so with that, I will close by. I'm sure that you have all the questions, and I'm very happy to answer your questions. This is a fantastic time for doing our kind of research. And if you're interested in working in my laboratory, I am very happy to recruit you, and you can definitely send me an email. So with that, thank you very much for listening, and I will see you soon in a more live interactive setting. Bye.